Hi everyone, my name is Sampath. I'm working as a researcher at Agriculture Agri-Food Canada. For the past 2 years, I have been using nanopore sequencing technology for my research. For this presentation, I'm going to give an introduction about what is nanopore sequencing, what's the impact of those sequencing technology for genome assemblies. Let me close this video so you won't get distracted. Learning outcome for this presentation is to make sure that you understand what is nanopore sequencing technology and how it works, and also to introduce about the importance of long-read sequence technology for assembly of plant genomes. Okay, before getting to nanopore sequencing technology, I'd like to give a brief introduction about what is the DNA sequencing generation. I assume that most of you know about what is DNA sequencing. The DNA sequencing is decoding the genetic information of our DNA. There have been various technologies have been developed on the course of time. This Sanger sequencing is the first generation sequence technology, which has been developed in the concept of chain termination method. The second generation sequencing is mostly sequencing based synthesis, like Illumina based, solid or 454 technologies. And third generation technology is the recent technologies, especially by PacBio and Nanopore and PacBio. Those are commercially available for now. These first generations are quite longer read size compared to the second generation, but the throughput, the depth of the sequencing you get is really low, which means that you have to pay more money to get more amount of sequence. On the other hand, second generation sequencing is NGS, next generation, also called as next generation sequencing technology, where you can get high throughput nature, but in terms of read lengths are really low, like less than 500 base pair. Recently, like last 10 years, there have been rapid improvement in third generation sequencing technologies. Nanopore and PacBio, those can sequence a read length of more than 10 kb average up to like 2 megabits, especially in the Nanopore sequencing technology. So in this presentation, I'm going to give an introduction on this Nanopore sequencing technology. And also want you to keep in mind that there are other technologies available for scaffolding. So once you got the sequences from this technology into quantic, then you put them into a scaffold to make a chromosome level assemblies. You may want to use this scaffolding technology, which is just a restriction based map, instead of using like genetic linkage map, which takes ages to develop with the populations. Before I'm going to introduce about nanopore sequence technology, I'd like to show you this less than 2 minute video, which will clearly depicts what I'm going to explain in this forthcoming slides. So let's watch it. This is the monoion database having flow cell. This is the flow cell. This is a DNA library. The DNA where you adapted your adapted sequence as well as motor protein, which then loaded onto a flow cell. Flow cell contain many wells or nanopores. For example, this is one of the nanopore where your DNA library is loaded. This purple helicases unzip the DNA. And the other DNA is entered into a nanopore. You can see the flow of the current. This process repeats. These are the nanopores. Okay, now you watch the video. The video shows about a nanopore, the purple helicase protein, which unzips the DNA sequence and make sure that it enter it into a nanopore properly. There is adapter molecule, adapter binds here, that is for the first instance to keep the strands together. So when the DNA is passes together, as a DNA are different molecules, different shapes, so it can uh, alter the flow of the ionic current. The current is caused as a DNA is negatively charged. When the flow is enter inside this nanopore, that DNA current is altered so that you can able to see that alteration. Then you capture those alterations as a graph. So this is called a squiggle graph. This was then decoded or base called to nucleic bases. This concept was around from 1989. Thus, Deemer is the one developed this idea, but coming into commercialization it took about more than like 25 years. In 2014, the first Minai sequence was available for uh, commercial, commercial available for every researchers, I guess. So this nanoprotein was then genetically engineered to make sure that it can able to pass the DNAs with the pace of like 400 base, at least 400 base per second, which makes them you can able to sequence in a high throughput manner. So overall, the concept of this number sequencing is to it works on this flow changes in the flow of ionic current, which leads to uh, have a different formation of different graph structure. So then which can base call two bases. There are two long-read sequencing technology available. One is nanopore sequence technology, and the other one is the PacBio. PacBio, as I said, the nanopore sequence also works on this ionic flow change that it was recorded in a graph manner, and then it base called. In PacBio, it uses synthesis model like polymerase to synthesize the DNA strand, which then when the fluorescent labeled nucleotide is incorporated, that you can see or you can record those the color change. So this color are then converted into nucleotides. This is how the PacBio works. Compared to Nanopore, PacBio is a kind of size limiter. You cannot sequence, you cannot get more longer reads in PacBio since it's sequencing by synthesis. However, in this case, a nanopore, it doesn't matter how big your size of the DNA, it just can go inside and it can produce. For example, recently, more than 2 megabase sequence has been generated. Single read sequence has been generated using nanopore, which is the highest by any sequencer. There's a famous sequencer for Minaya, which is very affordable to everyone, which is the cost like less $1,000. Another case, the attack bio, which cost about $500,000, which is really expensive. So it's, researchers can easily or affordably can develop their sequencing lab in their own lab. I'm going to introduce about few of sequencing instruments. It's not every instrument available from the nanopore sequencing. Here is Manayan, very famous nanopore sequencing instrument, which can hold a single flow cell containing 2,448 pores. This flow cell you can run it like 48-hour validity. You can run it in a single stretch, or you can do it on a multiple rounds. Each time 512 sequence channels sequence at a time, it can able to produce up to 10 to 25 gigabits. So once you used, you can just wash it and then you again load your library and then you can run it again. So that way you can run this flow cell for like more than one time. You wanted to have a more sequence of this genome, so better use it for 48 hours. There is other instrument called GridION, which is just a 5x of this flow cell. You want to increase the high throughput nature. You don't want like it takes about 2 days to complete. So you don't want to spend time on sequencing. So in the sense, uh, medium throughput can be achieved. For example, 10 giga data can be 100 to 125 giga data can be obtained from this flow cell in 2 days. There is the other one called Promethean. This sequencer has a different flow cells. It can have pores up to 12,000 per flow cell. So you can able to sequence 100 giga per flow cell. This is the flow cell model you see, which is used for Promethean, which means that you can able to sequence really high throughput sequencing for any genome. For example, in plants, you may want to sequence really high amount of Mm. High throughput means like high depth. You wanted to sequence, so it can be done easily using this Promethean, but it's quite expensive compared to other instruments. This might cost about $125,000. There is recently a Plangle, which is kind of testing its instrument, just has 126 pores. For example, you just don't want to waste your flow cell. Instead, just check your library quality and everything using this Plangle. This is a one-time use. So you can make sure that the library is really good. So you will not be waste your library. For example, sometimes you not obviously get 100 gigabyte library is not really good. So in that case, it will be really important. There are other instruments which is for like automate library preparation and other sequencing computers also available, meaning, but these are the major instruments you wanted to know for now. I'd like to give an idea of how and what's the price and how the sequencing kits look like. This is a so when you first time register and you can able to get all the materials in $1,000. For example, this is a MinIAM sequencer. This is a portable um, mobile-sized device with a flow cell. Flow cell it costs about $500 if you buy in bulk, and like it cost varies how much quantity you buy. And there are kits, library preparation kit, library washing kit. This is the importance of this nanopore sequencing is they have kits and everything, and it's very the steps are very mm, convenient to do, or it doesn't have to be like very professional, just only in terms of loading to this uh, loading library to the process. But most of the time, you get used to this technology very easily, and that preparation steps is not that huge compared to other um, shorted library preparations like Illumina ones. This slide summarizes the importance of nanopore sequencing technology. First, it has no capital or investments, so which means that every lab can afford to have this sequencer, and the library preparation method is really rapid, simple, yet effective, and the nanopore sequence can produce more than 300 kb an average based on depends on your library. Direct RNA and direct DNA sequencing is possible without any PCR. What we're doing is we are using sequencing by synthesis, especially Illumina case. And there will be high throughput and multiplexing also available. And direct methylome analysis, you don't have to do any separate sequence for methylome analysis. Just using this direct DNA sequence, we can analyze the methylome pattern. I'll show you in the forthcoming slides. There are major drawbacks in this sequence technology, as a new technology has been rapidly improving, like for chemistries or sequencing kits. When I was sequenced in a proposal, I used to get something like 2 giga, but now we in average we can able to get 15 giga. It was quite interesting. They have improved a lot of core chemistry as well as the sequencing kits. However, the major drawbacks, what I say is that read raw read error rates, which is about 2 to 15%, and this is really huge. You really don't want to have this, but this much error rate in your sequence. For example, in average, is yeah, gene can have like 100 base pair, 2 base pair, which is really huge. So that's you don't want to because of this rapid DNA pass through that nanopore it causes, and also it's not much. It also is just single rate based. There is other error called homopolymer error when it's just sequence produces the signal like a for the poly or poly T tiles. It doesn't convert our base call really well. And there is other one. It's not a really a major drawback I see. Like you need a really high amount, high multiple DNA, which is important for this. Here, okay. So using long read sequence technology, you got long reads. So what's the matter? Why you need? What's the importance in this of these long reads for especially the plant genome or plant genome assembly or annotation? For example, here on my left here, this is the tomato genome was sequenced using short read sequencing technology, mostly by less than 50 base pair. It's less than 500 base pair for more than 10 years with the effort from 300 people around the world. But yet it's not complete. It's lots of fragmented. This is what you see when because you don't know where to put these puzzles. As search rates, it's really there's a complexity. But when you have these puzzles are like bigger size, yeah, definitely you can able to put it into pieces together and make it more contiguous assembly. This is called more contiguous assembly. You won't have this complete assembly if you want to have them in a want to use that for further analysis and graph improvement. So this one you can do it in a month with 5 people, better contiguous assembly as the size of the fragment or reads are more than 10 kb. Even you can able to achieve 2 mega, like 200, 2000 kb. This is one of the examples showing how good the long read sequence technology. This is the chromosome 5 of the Brassica nigra genome, which is from our work. You can able to see these references in the reference slide. This is the black mustard genome, chromosome 5. This is done with short read sequencing, and this one is long read sequencing. You can see this, especially this collinearity means like those are basically from genes conserved. These white spaces are not available or not able to sequence with short read sequence technology. So, for example, we got about 20 megs are sequenced, especially this centromere or pericentromere associations. These greens showing that the depth of repeats. So when there is a lot of repeated in nature, so it's really hard to assemble. And you know that plants are really complex. Even 90% of the genomes are repetitives, especially for the maize or wheat. And you need this technology to comport those complexities and also polyploid nature. The other advantage of nanopore sequencing is you can sequence a full length mRNA instead of just fragmenting it. Using this, you don't know which isoform is come from this DNA, which is kind of awkward to use short read sequence technology when you just have this and we try to predict what is that mRNA is or what are the isoforms. But in this case, when you use long reads, you can directly sequence the complete gene, this complete 6 exon in a single read, which means that you don't have to worry about assembling those short reads. And other importance of this Nanopore sequencing or long read sequencing is its direct methylome analysis. For example, you can see this graph, Spiegel graph, shows the C's when the methylated C and the methylated C and the unmethylated C showing different pattern, where it can just with the direct native DNA sequence, you can achieve this. But in the case of when you want to do with short read or bisulfite sequencing, you have to prepare a different library, which is does the bisulfite conversion, and then you do the sequencing. So those steps are can be eliminated by this long read sequencing technology. Nanopore are as well as PacBio also able to predict the DNA methylome using the DNA sequences. As I said, there is the one that Nanopore has some drawbacks. I found the most important one is the long read, raw read accuracy. It has about like to 10% or 15% error rate. So this is what you see when you read the raw or uncorrected Nanopore reads or the Illumina reads. Illumina reads are 99.999% highly accurate reads compared to Nanopore reads or long reads. But you can correct those reads using those when you have a high depth, also using that short reads. So you map the short reads and you can eliminate or you can minimize or eliminate those error rates, or change it, or just correct those rates using that. There are programs available. Okay, summarizing my talk. So in this talk, I hope I have explained about what is nanopore sequence technology, how it works, and the principle of that change of flow of the ionic current, which causing the changes in the graphic pattern. So then can be based on. It does the sequencing without synthesis, unlike any other sequencing technology available. That's why this sequence technology used in space as well. This nanopore rates helps to assemble genomes with really high complexity nature and to bring into a nearly continuous full length assemblies. And nanopore sequencing technology can have a high error rates, but it can be managed with the tools available, especially when you increase the read depth, or also when you when you use that short read technology. For now, you have to use this method, but in the future, there have been development is going on, like using double sensor, which means that when you predict, when you get the signals, highly accurate signals, signal graphs, then can be converted into high quality basis, which way that the error rate can be minimized or eliminated. Direct RNA sequencing and the methylome analysis is really a plus for this nanopore sequencing. So I definitely suggest like whoever getting into sequence technology, this is a really good technology to use. This nanopore technology not only for that, this the whole genome. You can also do the PCR sequencing or like a sequence enrichment analysis, whatever. And it's also multiplexing. There have been technology for high throughput genotyping. It's still on the road. Okay, so there are further references and this is for further reading. You can go through this. There are also good paper from Nature Biotechnology. I also have worked on this for the past 2 years. So we have a bioarchive available. So we hope to publish this work soon in Nature Plants. Nanopore, there's a good paper about if you want to have a practice on how nanopore sequencing sequence or the softwares and the codes, and there is a very good this paper as well as the GitHub page. So make use of it. And if you have any further questions, yeah, you definitely feel free to contact me. I really like to thank Dr. Sanjay, for giving me this opportunity to share my our experiences here. With that, I'm going to stop my video and I'd like to say goodbye. Stay safe and stay healthy, everyone. Thank you.